In this video, we will study about the genetic basis of the oral cancer. Genetic basis of the oral cancer. Basically, the molecular pathogenesis of the oral cancer, most commonly the squamous cell carcinoma. So, this is the cell membrane of the oral keratinocyte. So, as we know that the cell membrane of oral keratinocyte have a receptor, we called it as the epidermal growth factor receptor, where the epidermal growth factor attaches. So before discussing the pathogenesis, we will study about some normal concepts. So whenever epidermal growth factor attaches to this receptor, so what happens is that there is an activation of the RAS oncoprotein, which is made by the RAS oncogene located in the inner side of the cell membrane of oral keratinocyte. So normally what happens is that RAS oncoprotein is attached to the GDP. That is, it is in the inactive form. But whenever epidermal growth factor attaches to its receptor, it gets attached with the GTP. That means it is in the active form and it stimulates the cell to proliferate. So there occurs a cell proliferation by a method. So after the cell proliferation is over, there comes the GTPase and it again converts into the inactive form. So GTPase is basically the function is controlled by the GTPase activating proteins. So how the excessive cell proliferation leads to the oral cancer. So there occurs a mutation in the RAS oncogene. So if mutated gene will form the mutated RAS oncoprotein. So mutated form of oncoprotein will bind to the GAPs, that is GTPase activating proteins. So that's why GTPase will not be in its function, and RAS oncoprotein will remain actively bind to the GTP, and there occurs a excessive proliferation. So how the cell proliferation is controlled by the other genes? So first is the RAS oncoprotein. We know that the RAS oncogene makes a RAS oncoprotein. So how the proliferation occurs? Because the RAS oncoprotein activates the kinase cascade, and this protein kinase, mitotic activated protein kinase, activate the cellular MYC, which is again an oncoprotein, and it activates the cyclin D in the nucleus. That activates the cyclin dependent kinase. So now cyclin D kinase is activated. So we have a P retinoblastoma, that is a retinoblastoma protein that is found by the retinoblastoma gene. So what happens is that normally it is attached to the epidermal factor, but cyclin D kinase causes a phosphorylation of the P retinoblastoma protein. So now epidermal factor is free. So epidermal factor, when it is free, it is in the active form. So in the active form, it will lead to the transcription of the various proteins located in the DNA, like that of the pCNA. It stands for the proliferating cell nuclear antigen. Proliferating cell nuclear antigen. It is one of the transcription protein. It is one of the transcription factor or the protein located in the DNA. So this is a DNA, and it is surrounding the DNA. So it proliferates or it stimulates the DNA replication, and there occurs the cell division. So, okay, before coming to next concept, we will again study some terminologies or some normal proteins like P53. It is very important gene. It is a tumor suppressor gene which makes a P21, and we have two another genes like BCL2 and BAX. BAX is responsible for the apoptosis, and BCL2 is an anti-apoptotic gene. So we have an enzyme called as a caspase 3, which is responsible for the apoptosis of the cell, or apoptosis or cell death, or the apoptosis of the cell. That is a programmed cell death. So when P21, the function of P21 is to activate the BAX gene, that will lead to the apoptosis of the cell. So P53, very importantly, it blocks the BCL2. So caspase 3 will remain active and it will stimulate apoptosis. So p53 also controls the DNA damage. So what happens if p53 gets mutated? So the DNA damage, that is the RAS oncoprotein mutation or RAS oncogene mutation, will not be carried by the p53. So apoptosis of the mutated cell will not occur and there occurs an excessive proliferation of oral keratinocyte leading to the oral cancer. So 80% of oral cancer show the mutation in the p53 gene. So loss of the normal p53 gene leads to the increase in the cell proliferation even in the absence of the epidermal growth factor. So this is the oral keratinocyte. This chart is given in the Schaeffer's. So when epidermal growth factor attaches to its receptor, it activates the RAS oncoprotein and there occurs the activation of the kinase cascade. So this is the nucleus. So there occurs the activation of the cyclin D kinase and we have two genes, retinoblastoma and the p53. So normally the DNA damage detected by the p53, but when it gets mutated it will not be detected by the p53. So BAX is a apoptosis gene and it will causes a apoptosis of the mutated cell. So this was given by the Sugarman and Savage in the year 1990. This whole concept or the pathogenesis, molecular pathogenesis of the oral cancer, basically squamous cell carcinoma, basal cell carcinoma and the malignant. The etiology is different but the pathogenesis is almost same. So 80% of oral squamous cell carcinoma shows a mutation in the p53. p53 it is called as because it makes a protein that have the molecular weight 53 kilodaltons. So that's why it is called as a p53. So this was all about the molecular pathogenesis and in this we have the 3-hit concept. 3-hit concept theory, that is first hit occurs at the genetic level, that is genetic predisposition and where the one allele is mutated, and second hit when the second allele of the p53 gene is mutated in the early life, and third hit in the later life. So both the alleles get mutated and p53 will lose its function and due to the environmental factors there will be the